Okay, so good morning, everyone. We want to just thank you for giving us the opportunity to tell you a little bit about our collaboration with Andrew's lab, and it's entitled the Cardiac Gene Wiki Project. So this is a team effort by the names listed here. The mitochondrial protein annotation data that I'll show you has been accomplished in large part by Anders and by Jennifer, and the tools have been developed by Tevfik. So let's see if I can get to the next slide. There we go. So basically the premise we're operating on here is that we are cardiac scientists doing biocuration. So we see this great portal, GeneWiki, developed by Andrew's lab, and we see an opportunity to elevate both the quantity and quality of cardiac knowledge annotated within the pages of Wikipedia. So the way we do this is to break the cardiac cell down into functional subproteomes. So these are sort of manageable protein gene families within which meaningful relationships can be annotated. So we have expertise in our group in cardiac mitochondrial biology and in cardiac sarcomeric biology. So the great value in this approach is that it enables rigorous validation of the scientific content by us or the domain scientists to ensure knowledge of high accuracy, depth, and breadth, which then gives the annotated knowledge a high degree of usefulness and reusability. So we selected 5 subproteomes from the human cardiac mitochondrial proteome for improvement. And these are metabolism, there are 312 genes, oxphos, 101 genes, proteolysis, 58 genes, redox, 31, and apoptosis, 54 genes. Basically, our protocol for improving articles is to first create pages in BioGPS for those lacking stub articles, and then database content is automatically presented in the protein box using the PBB template developed by Andrew's lab. Next, we do sort of intensive literature searches and populate a list of journal articles relevant to the gene or protein, and then we organize this information into sections of introduction, which includes basic information, and then a little more detail outlined in structure, function, clinical significance, and interactions. Then we write each section, incorporating key data into understandable and concise text. Then we establish wiki links between the articles, between, for example, diseases, concepts, etc. And then we put a significant amount of effort into comprehensively citing high-quality references from PubMed to facilitate knowledge access. So here's a little summary on our progress to date. We have added roughly, well, first of all, we've improved 800 sections across 200 mitochondrial gene protein pages, and we've added roughly 1,600 references and 3,500 links. We've added about 1,100 kilobytes of content, and this has resulted in a 25% increase in complete pages. And incomplete articles have been reduced from 2/3 to about 1/3. So basically, in reference to the entire human genome, we've annotated roughly 1% in this manner. So here are pie charts just showing the initial status versus the current status. So initial scoring of the articles showed that only 1% of mitochondrial articles could be deemed complete. So we worked over the course of a few months to improve these articles, and we were able to increase the complete articles from 1 to about 26%. So the mostly incomplete articles were reduced from 46% to 26%, and missing articles from 22% to about 15%. So basically 31 pages, missing pages were created, 169 incomplete pages were improved, 200 total pages were completed, 800 sections improved, and then I've already shown you these data, over 1,600 references added, 3,500 links added, and over 1,100 kilobytes added to Wikipedia. So looking at it in terms of percent improved, it sort of stood out to us that both clinical significance and interaction sections were markedly improved by 20 and 18% respectively. So since the start of our project, Tevfik has developed a couple tools that have aided in our curation efforts, and these are shown in this workflow figure here. First, the article analysis tool. It's basically a quality assessment tool that analyzes and discovers incomplete articles in Wikipedia. And then the curation tool is used to search PubMed more efficiently prior to curation. And then these two tools will be discussed in the following slides. So the article quality assessment tool was developed again to automatically evaluate GeneWiki pages to determine the word count of the sections within. Basically, the user inputs a list of Uniprot IDs, which are then mapped to the corresponding Wikipedia page by the GeneWiki portal mapping service. And then the tool retrieves articles through the Wikimedia API and determines the word count in each section of the article, including introduction, structure, function, clinical significance, and interactions. And then the word count of each section is normalized to provide basically a score that's ranging from missing to complete. And then this is presented in a user-friendly spreadsheet for the user. So then the curation tool, it provides a web interface that parses user inputs and applies filters and constructs API calls to the PubMed Entree API. So users input search terms, including, for example, genes, proteins, metabolites, chemicals, or pathways in the left column. And then the crosstab search terms such as organ type, tissue type, or disease in the right column. And the PubMed API is queried using the left column as the main search terms and the right column as conjunctive search terms. So then the tool populates this contingency table, it basically indicates the total number of articles for the main search term as well as that from the crosstab query, with links to the search results on PubMed. And you can access this tool at this website down here. So basically, these are just two straightforward tools developed that have aided in our curation efforts by making the process less tedious and more user-friendly. So I'd like to acknowledge everyone that's played a role in this process. So as I said, Jennifer and Anders have spearheaded the mitochondrial efforts. Tevfik has developed the tools. We have recruited our colleagues to help us annotate the mitochondrial proteome, Ding, David, Edward, and Jessica. And of course, we're grateful for our collaboration with Andrew's lab. And with that, short and sweet presentation, I'll be happy to take questions.
Thank you, Sarah. Are there any questions?
Sarah? So in a database approach, usually, in a database-oriented approach, you tend to annotate now many resources, the kind of evidence. So whether it's directly from the literature or author inference or so. Do you have anything like this? Can you do anything like this in GeneWiki, or is it just everything that you put in has the literature reference, so it's direct literature evidence, and that's it, and you don't need the distinction because it's all from that kind of evidence?
Yes, the latter is sort of our approach. Everything is heavily referenced. We try to do this as comprehensively as we can, so we formulate sort of concise concepts. That's how we write the text. And then we heavily reference each of those concepts. That being said, the protein box has a significant amount of information in it, such as, chromosome, gene information, Uniprot, PDB, excuse me, all of this type of information from other databases, and that's presented concisely in the right column of each page. But as far as our role, ours is basically unstructured. We write unstructured text and then heavily reference it with the literature. Is that answering your question?
Yes.
Okay.
The other
any suggestions?
I've got another question, which is, well, ultimately it's more about reporting, but can you and how can you pull out from GeneWiki those entries which have been annotated in the context of this program.
I'm sorry, I couldn't hear the first part of your question. Could you please repeat it?
So you are now annotating directly in GeneWiki. Is there a way, is there a tagging, or do you have a list of what you are doing so that, let's say, at report time at the end of this grant, you can pull out all the entries which have been annotated specifically in the context of this program, or is it just we are happy to improve the overall content of GeneWiki and that's it?
Okay, so we have a direct record of all of the proteins that we have annotated in this manner, but it's only as sophisticated as an Excel spreadsheet. I think what you're asking is, do we have a way to, is that what you're asking? I'm sorry.
Yes, so basically, if you, just to give you an example, in InTact, we are annotating protein interactions.
Mm-hmm.
We are tagging protein interactions often with one, sometimes with multiple tags, for which, let's say, scientific data separation, for which funding they are relevant, and also who has done them based on which money, so that we then can easily report to the funding agencies.
I see.
Content of InTact, but specifically with your chunk of money, we have done these.
I got you.
In this disease area.
Yes, we do not have anything like that in place, but that is definitely something we have discussed, and that is a very helpful suggestion.
I think that is something which should be somehow possible, and probably you can talk to Andrew also how this can be technically implemented. But I think this is very nice to show, well, ultimately what has been done for the money, but also to really pull out what are the genes which you considered important. So that's probably also not possible right now to say, give me all the proteins from the mitochondrial protein, right? So I think something like this would be very valuable to do.
Thank you. I 100% agree with you.
This is Andrew. So just to follow up on actually both of Henning's points, I think Henning, you raised obviously great ideas. Since you brought up the intact curation effort, right? I mean, I wonder if there aren't possible synergies there, in the sense that you're rightly pointing out that almost everything that we do in the context of the GeneWiki, with an asterisk that I'll get to in a second, but almost everything we do is unstructured knowledge, right? We're taking free text and we're summarizing it into more free text, which I think is valuable, but there's also value in the structured knowledge that databases like InTact and gene ontology annotations and so on provide. So I'm wondering, right? I mean, because the cardiac experts, Jennifer, Andrew, Sarah, Edward, so on and so forth, they're already reading that literature to summarize it, and I wonder if we provided them with some sort of annotation guideline, whether or how difficult it would be to just take the next step and, you know, generate some candidate InTact or Go annotations that then could be counted in more traditionally recognized ways as well.
Yeah, I think that's a wonderful suggestion, and that's something we should definitely talk about, is how to implement that. Because absolutely, if we're going to all of the trouble to read all of these papers, organize the papers into, you know, structure, function, clinical significance, interactions, and this is all very tedious and time-consuming, and we should really be getting the most bang for our buck, if you will.
Right. Go ahead. Any comments?
We have a similar way of interacting with Unit. where sometimes Unicode curators will just forward the paper to Impact and they say, oh, there's a lot of interest in direct.
I'm sorry, there's a lot of background somewhere.
Yeah.
I will try one thing.
Yeah, it's really.
Can people mute who aren't talking, perhaps? And then maybe Henning can try again.
Salva, who is Salva? There seems to be a lot of noise coming from Salva.
Great, perfect.
Okay, so with Uniprot, we have two modes of interaction. One is Uniprot curators will come forward relevant papers with lots of interactions to the intact curation team. And then we will just curate it. And the other way is that the few Uniprot curators have intact curation training, and then they will directly curate into intact. They tend to not like the deep curation which intact does in terms of annotating the experimental system in detail, like which were the, let's say, the tags, what was the precise technology which was used. And so the Uniprot curators tend to do what we call mimics compatible, so a kind of lighter curation. But indeed, this is what Andrew termed stub or draft curation already, which then only needs to be deepened by the intact curator. And a lot of the work is actually in figuring out what are the precise Uniprot identifiers in a paper. And then picking up from there, or just leaving it like this, is something which we could consider, yes. The other point, again, thinking a bit, what we could do easily is once we have this list of, let's say, the mitochondrial proteome as annotated in this project, then we can just, with a handful, or we can just have a hard-coded link, which then goes with this list of Uniprot identifiers and can give at any point the corresponding interactome directly from intact. So that is a SNP, basically.
Okay, great. Wonderful. Maybe we can schedule a follow-up call, all of us together with Peipei, and we can discuss how to proceed. Thank you. These are all wonderful suggestions.
Henning, can I ask another question about the intact curation? You know, as you think about the effort required to go from, you know, draft/stub curation of interactions to the deep interactions, like how far is the stub getting you there, right? I mean, is it you know, is half the battle finding the right articles, and in which case, once you know somebody says, "Hey, here's the right article," you know, that's you know 60% of the value, or is it like, well, you guys are swamped with articles, and the deep curation is what's the most time-consuming part anyway?
So finding the article is not a problem at all. So that's probably somewhere between 5 to 10% of the work. The more relevant part of the is, which would be part of stub curation, would be identification of the precise entities which are interacting. So if you go, that's also why text mining for us very often doesn't get us very far. It's cheap to get the relevant papers, and text mining traditionally is not very good already at sorting out which species is actually being talked about. The gene name is usually easy, but the species is something where text mining tends to be quite weak often. So that's why we make only limited use of text mining. But if that is done as by a human as stub curation, that already gets us a significant part down the line.
And just to confirm, right, I'm trying to think of going from a protein name or a gene or a protein name, let's say, plus species, and going all the way to a Uniprot identifier. Now, I imagine it's a great source of frustration for you that many articles might not even report the UniProt identifiers? Is that perhaps the case?
Yes, so if the UniProt identifier is there, obviously it's easy, but what is unfortunately a frequent case is that the article is talking about the mammalian LCK clone, and then we have to figure out what actually was used, which species, and then we sometimes have to write to the author, and if we don't get an answer, then we have to register that paper as not curatable.
Got it.
So therefore,
that
part which is already being done now of, yeah, A, but the precise A interacts with this precise B, that already would be, yeah, a significant part of the work. So that's why what you suggested might be actually an interesting approach. And again, we are doing something similar already with UniProt.
Right. I'm just wondering. Right. I mean, on the UCLA team side, it is still a pretty lean effort. So I'm wondering, you know, how deep they're going to be going in terms of emailing authors and things like that. I think, right, the ideal world is that the thing that the literature reading that they're already doing would synergize with the you know, the workflow, but yeah, I'm just, you know, not sure in my mind how far we should, you know, the UCLA team is going to want to go down that track.
Yeah, that's what I indicated. It's why the collaboration with UniProt is on the level of this so-called light curation, and it's really very much when it fits. So it's clearly not something which is done systematically on every paper, and I would imagine if we do something like this, it would be similar, where, oh, here's a clear clinically relevant interaction. This should be an intact. I've already done most of the work, so I just go to the intact interface and put it in, or at least I forward the article and intact does it, while if it is, oh, this is a messy article and it's not really that relevant, then we just wouldn't do it.
Got it. And one quick follow-up question. Am I guessing that the majority of what UniProt does in terms of light curation, more of the individual articles studying individual proteins as opposed to sort of large-scale proteomic screens, which I'm guessing Intact has pipelines for getting, you know, 95% of those anyway?
That's correct. So UniProt is clearly more focusing on the small-scale papers, but they occasionally also import proteomics papers. Now that is done in a fairly well-organized way. It was done for quite a while very ad hoc, and there was quite some pollution of UniProt, for example, with phosphorylation sites, which then, from mass spec, which then turned out not to be really reliable, and they were just old experiments. That is now done a bit better. Intact has, I would say, a good coverage of large-scale experiments because a lot of the bigger players in the community are now in the habit of sending the data to us, or we will pick it up. But on the small scale, we have probably many more small-scale papers than large-scale papers, but the coverage is much lower on the small-scale papers. And especially to have this sort of pre-filter, this is something highly clinically relevant in a collaborative project. That would already be interesting.
Makes perfect sense. Yeah, so I mean, you know, as I think about, you know, what the UCLA team, right, has done and where we could go forward, I mean, obviously it's a great amount of effort and highly valuable. I think we should have you know, I think one of the ways we could push this forward, right, is that, you know, use cardiac gene function as sort of a prototyping tool for different communities to use, right? I mean, you know, you guys contributing effort, right, is again very valuable, but it doesn't have a multiplier effect. You know, and we want to get that multiplier effect in place where it becomes, you know, big data tools and applicable to other communities. And so I guess what I'm thinking is, okay, well, we have some sort of basic, some tools that Tevfik has built to help that manual curation process. But I wonder if, right, we shouldn't push a little bit further into more structured annotation. So, the cardiac community would be sort of a domain-centric starting point. Okay, here are tools where you can annotate in the GeneWiki. Here are tools that you can annotate light annotation into intact. Here are tools where you could do light annotation into gene ontology. And because I will say, from the gene ontology side of things, so I happen to be on their scientific advisory board, and I don't think that they have very systematic and useful ways of interacting with domain experts. They really emerge off of one-off efforts that just happen to get funding. But creating some tools that allow, again, domain experts to interface with curation efforts across the spectrum, I think could be very valuable.
Great, thank you. Yeah, we couldn't agree more, honestly.
So I have one other, I guess just comment or maybe a discussion point off of the first issue that you raised, Henning. Right, so the issue, I think I've raised it before, but right, we've been doing more and more stuff with Wikidata. And Wikidata, so Wikipedia is to free text what Wikidata is to a database. So it's a community-contributed, community-maintained, structured data database. And it's getting more and more traction. It's supported by the Wikimedia Foundation, the same people that support Wikipedia. There are increasing links between Wikidata and Wikipedia. The idea is that Wikipedia is withdrawing data within Wikidata. So one of our initiatives through, not the BD2K, but through our R01, is to import as much biological data into Wikidata. So genes and gene annotations, protein annotations, diseases, drugs, things like that. And the advantage of being in Wikidata, just like the advantage of being Wikipedia, is that the data, that the resource is essentially scopeless. Like Wikipedia, right? You could go from articles about a gene to an article about a disease, to an article about anatomy, to an article about development, right? To an article about epidemiological data and so on and so forth. And in the same way, Wikidata would be scopeless in terms of how far you could, just do your structured data queries. So I don't have a specific thought or suggestion here, but I just wanted to reemphasize, especially to Henning, like that's one area where we're moving forward, beyond GeneWiki just being another unstructured data resource, but pushing into wikis and structured data.
That's wonderful. Another sort of thing we've discussed too to sort of structure some of these biological relationships within the text is to insert semantic wiki links into the text, which are not, of course, queryable within Wikipedia, but then we can use like GeneWiki Plus, to query these relationships. So that's something we want to also implement.
So I would suggest, Sarah, actually, that that we don't do that.
Okay.
As much as idea, and it was, it just never took off, and it never got traction. And on top of that, Wikidata should be the right way of encoding.
Okay.
So again, we should continue to touch base on our Wikidata efforts, because our goal with the Wikidata project is to define the relationships between genes and proteins, between genes and proteins and diseases and drugs, and so on and so forth. That's exactly the use case for the semantic.
Okay.
When we proposed semantic wiki links, Wikidata just didn't exist yet, and there was no.
Okay.
So that also is, hits along this idea of light curation in another domain area. So the way we can imagine that working in the future would be, we might be able to create a light annotation interface for these sort of relationships. And, you could imagine this, right? When you annotate a protein-protein interaction according to the according to the intact, light annotation template, right? That obviously sends a report out to intact, and then that might also spawn an entry into Wikidata.
Mm-hmm.
But similarly, when we have types of relationships that aren't handled by any database within our consortium, so for example, protein or gene-disease relationships,
mm-hmm.
It would be great to have some sort of tool
mm-hmm.
where you guys can enter that into Wikidata directly,
mm-hmm.
and it doesn't have the visibility yet of a Wikipedia page, but it's really valuable, and it is amenable to counting and statistics and metrics of the type that Henning was
describing. Yeah. Yes. Okay. Absolutely.
So there was some talk earlier about engaging sort of broader communities of students. Is that something that you guys have pushed on at all?
Yeah, so that's something in the pipeline. We haven't, right now we're sort of, our focus is to gain some credibility that we can actually, make high quality pages, and, a lot of discussions have gone into that, and as you said, it's taken a lot of work. But that is sort of our next, that's in the next phase, is engaging these students who are highly enthusiastic, but a lot of times have a low amount of domain knowledge, but, finding ways to give them an opportunity to contribute. So, but we haven't placed a large amount of effort into that particular part of it yet.
Okay, well, I think you're absolutely right that it's essential that you all individually understand that process, because if you don't understand the process, then obviously you can't create effective tools for other people to use, much less for you to use yourself. But I think we're on the same page that trying to move into tool development space is something that is or continue the move into tool development space is something that we want to do within the context of BD2K, right? BD2K isn't just about, supporting a small manual by iteration effort. It's about developing tools that generalize big data in general.
Right. Yep. Thank you. Anybody else? These have been awesome suggestions. And we can sort of join heads to make a clear plan of how we're going to approach this.
Are there any other questions for Sarah or any discussion points? Okay, if not, thank you, Sarah, for the presentation and initiating great discussion. Next week we'll be having presentations and collaboration discussions about biology PS and lygene.info. Yeah, but yeah, if anyone has any questions, we'll be online until 9. But yeah, thank you everyone for attending today's conference.
Great, thank you. Bye-bye.